I'm pretty sure.
We see you.
It's recording, right? Yeah. So it's recording. Oh, that's really awkward. Okay. And so then I share my screen, I think. Okay. Hi, everyone. I'm learning how to use Zoom. Okay. Present. Look at that. We did it. Woohoo. Okay. So, hi, everyone that's here. We are doing, do we just sit? Do they normally sit or do they stand?
Where would I stand?
I don't know. So you're gonna click through. I'm gonna go sit on the other side. So today we are going through, or this week we're doing DNA replication error correction and DNA damage and repair mechanisms. Woo!
Emily, you ran me over.
Fine.
I'm sorry. Okay.
You'll be fine.
So we're just gonna go through. We have different sections. Can you get closer to them? I don't. You know what I'm saying? Like, I just know about that. Oh, perfect. Okay. So we're just gonna go through slides. We have time for questions. Like we have slides that you can ask questions and stuff. So we're just gonna start going through. Feel free to stop us at any time, but every few slides, we have one that says questions where you can ask questions. So this is a meme. I didn't make this. So if it's not funny, it's not on me. But Laura also didn't make it. It's not on her. She doesn't understand it.
Who made it?
Huh?
Who made it?
I'm not going to expose someone like that. Tina. Okay, so we're going to start with DNA polymerase. This picture is actually really helpful. Piece of advice. There's actually a lot of really great pictures on here. So DNA polymerase proofreading. Proofreading means that instead of the normal amount of errors that we would make without proofreading, there's only about one mistake in 10 million nucleotides. And there's the polymerase has an additional domain or subunit with the DNA endonuclease activity. What does an endonuclease do? Anyone?
It cleaves the wrong nucleotides.
It just cleaves DNA generally. So we have, our polymerase has an endonuclease domain or subunit. And it moves the incorrect nucleotide to the endonuclease activity site, which is where the enzymatic activity is happening. So the endonuclease has a 3 to 5 endonuclease activity, which kind of goes with why DNA strands are synthesized 5 to 3, because we kind of catch it. Does that make sense?
There's also a video they can watch after the show. I said there's also a video they can watch the show, but that's later on. I'll show you later.
Okay.
But you want to?
Yeah, let's move on. Here's a picture. We're making it, and then we're proofreading it. So this is our polymerase, and then this is our endonuclease, and it's showing that our endonuclease attached, and it's like, let's stop. So that's that. Do we have any questions on DNA polymerase proofreading? No? Good. Awesome. I think everyone really just does, like, I feel like everyone hates me. I don't know why. Like, I feel like all of you are like, you're doing a terrible job. I'm so sorry. Okay, DNA damage and repair mechanisms. This is where we get into the fun stuff. There's a lot going on here. So here's a cute little video that I actually think is really helpful. So we're starting with mismatch repair. So I don't think it's relevant, but the error is about 1 mistake every 1 billion nucleotides added. So this is post DNA replication repair pathway. So proofreading is happening during replication, right? So this happens after replication, and we search for and fix a base that has mismatched. So that's basically, if we're matching with a T, if it's anything other than an A, that's a mismatch. And it's important that we have to differentiate between the newly synthesized strand and the original strand, because if we change the original strand, then we're messing up the original DNA. So this is a really important step to know which one's newly synthesized and which one's original. And we will get into the bad outcomes if they are not differentiated. So if no repair occurs, then this is kind of complicated, so I'm going to go through this a little more in depth. So if no repair occurs, right, then we have the template strand didn't get changed, right? So in this case, the template strand is still an egg. So it's still connected to a T, and so this is fine. But the G over here is supposed to be a T. This mismatches with no repair. So this is a mutated strand. So if we have no repair, we have one normal strand and one mutated strand. And that's how we get that. If a repair occurs on the template strand, both daughter cells are messed up. This is what I was saying, because not only did we get mismatch here because we changed this. This is the template strand. So I changed that to a T when all along it's supposed to be a G. So instead of this being a G and a C, it's an A and a T, so that's messed up. This was supposed to be a C and we didn't fix that because we screwed up the other strand. So now this is also an A and T. These are both mutated. So that's what happens if we mess up the parent strand, which is why it's important that we find the synthesized strand. And if we repair on the newly synthesized strand, then it's totally fine because we put the correct nucleotide back in and there's no mutation on it. Does that make sense? Yeah, should. And then the MMR mechanism, this is how we're actually doing this mismatch response. So the regions are lesions. That's what they're going to be referred to as. So there's proteins in these organisms where we can find these lesions, and we have to identify the newly synthesized strands to find these lesions, as we said. Different organisms do it different ways. So prokaryotes, E. coli is our favorite one to look at, is hemimethylated after replication for a short period of time. This means one strand is methylated, the other strand is not. Eukaryotes, we don't super know, but we think that the newly synthesized strands have more nicks in them, and we can recognize those nicks, which are the little cuts in the backbone. But that's just a hypothesis, so that needs to be studied more, like a lot of things in bio. And then we will go into more specifically. After we identify our newly synthesized strand using the methylation, we have to remove the area around the newly synthesized strand. So usually they take out a whole chunk around the strand at the DNA mismatch location. So we know that helicase has to come in and separate the template from the mismatched strand because we're not doing a double cut. We're just cutting the newly synthesized strand. And then the endonuclease, like we said, cuts it and we remove it. And then we have single-strand DNA binding protein come in. And we went through all of these in class, right? So single-stranded DNA binding protein stabilizes the single-stranded DNA. Our sliding clamp and DNA polymerase recruited again, we fill in the gap. And then we need DNA ligase to come in because if we opened it, we got to close it. And in prokaryotes, the new DNA strand is also methylated. So do we have any questions on that?
I'm going back like 2 seconds. This slide.
Yeah. No, Isaac, I can't do that. I'm sorry.
He's not wrong.
Oh my God.
Isaac and I go way back.
Yeah, we go way back.
I still don't like her.
Oh, you know what? Me neither. So it doesn't matter.
This is going to be recorded and put on YouTube.
It's okay. They can all know our beef.
Okay, awesome, love that. Okay, are we good?
Yeah. Thank you, Emily.
No problem.
Oh my God.
Okay, so this is another meme that if you like cats, maybe you'll like it. Common types of DNA damage. So why we need to be fixing it?
Mutation?
Huh?
Mutation?
I understand that you don't think it's funny, but so we got base-specific DNA damage. First, we're looking at depurination. So we're looking at our purines, right? So our purine base is removed due to spontaneous breakage of the sugar-base covalent bond. So the sugar-base covalent bond is obviously the bond between sugar and the base. And we spontaneously break, and obviously that is a problem because then we suddenly don't have the correct, we're not an adenine or a guanine because we don't have the base. So it's an A basic nucleotide, and it occurs about 5,000 times per cell per day. Also, it's how to remember purines versus pyrimidines. I don't know that. Does anyone know that? Yeah, I don't know that. I just memorized it. Purines are adenine and guanine and pica. So pyrimidines are cute. And they're cytosine, uracil, and what's it on? That, yeah, that one. I don't know why I just wanted to say threonine so badly. Okay, so this is depurination. It's a purine, so that's in the name. Teaching. Thank you. And then we have deamination, which we change our cytosine to a uracil. And that's a problem because one is in DNA. No, that's not true. Uracil, one's in both, and uracil is only in RNA. Okay, you want to present? So this is a spontaneous oxidative reaction, which means it will just happen randomly and doesn't require energy. Changes our CN bond to C double O bond right here, which changes our cytosine to our uracil. Apparently this occurs about 100 times a day. So this is pretty frequent. And then we have pyrimidine dimers where we end up having covalent linkages between usually two thymines together. So here's my two thymines. UV light can cause this. It hits the two thymines together, and then we get covalent linkage. And obviously, we know that covalent bonds are super strong, and that causes a problem with how we're supposed to be structurally functioning, because we're going to distort the helix if we start adding covalent bonds. So high energy wavelengths, like I said, UV light, and that is how that happens. Any questions on that one? Any questions on any of those three? The difference between pyrination, the second one, and the third one. Yes, no, maybe so. Good. Okay. Same meme. I said stop. Same meme because we couldn't find a new one. Primary repair mechanisms. So we just found out how we damage the DNA. Let's figure out how we repair the DNA. So for base excision repair. So this one.
After you're done, I want to play a video that shows them like how it happens. Okay.
So base excision repair, we can use this for depurinations and deaminations. So we can't use this obviously for the covalent linkages we were talking about, but this one we can get more into it, but the way I like to think about it, we literally cut out one base, take it out and put a new one in. So this is super specific. So deamination, well, for both the endonucleases, phosphodiesterase cleave the phosphodiester bond over here. This is our backbone on either side of the nucleotide. In deamination, we use the A basic nucleotide we can identify where the uracil and sugar bases, and we can cut that out. So that specifically happens for deamination, but for both, this is normally what we do. And then the sliding clamp and DNA polymerase can come in and just fill in the missing nucleotide. So this is very specific. And then we know if we use endonuclease, we have to use ligase to seal it back up. There's more to it, I think. I think there's another one that has pros and cons. Nope, sorry, go back. So now Chi-Chi's gonna switch over. Did you email it to me?
Yeah, but you're recording, so it's better doing your laptop.
Yeah.
Where's your email?
Go to the bottom. A little mail. You're really gonna expose my email right now.
It's okay. You'll be fine.
Press the back arrow. That's the back arrow.
I don't know how to work whatever this is.
GG.
I'm sorry.
Oh, let's not look at my statement for my. We good. I don't need to be exposing me like that today. It's embarrassing. He can't get cold. He's gotta run back from the store so he doesn't get cold. Oh, so this is a burr video.
Some damaged DNA bases are repaired by a system called base excision repair. Base excision repair works differently from nucleotide excision repair, which clips out a section of DNA containing a pyrimidine
Spoiler alert.
or other defect.
You're gonna watch
to
the video.
replace its DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil. Uracil can also be incorporated erroneously into a strand during DNA synthesis. If uracil forms in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine, uracil forms a base pair with adenine. During DNA synthesis, DNA polymerase would incorporate an incorrect nucleotide into the growing strand. A few other types of damage are also repaired by the base excision repair system. For example, when adenine becomes deaminated, it forms an unnatural base called hypoxanthine. A mutagen can also damage adenine by alkylating it, converting adenine to 3-methyladenine. Normally, adenine forms a base pair with thymine, but hypoxanthine in its place is mutagenic because it forms a base pair with cytosine. 3-methyladenine is even more damaging. When it is encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair employs a battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA glycosylase, 3-methyladenine DNA glycosylase, and uracil DNA glycosylase recognize hypoxanthine and 3-methyladenine and uracil respectively when these bases are present in DNA. The glycosylases cleave the bond connecting the damaged base to deoxyribose in the phosphodiester backbone. The result is an intact phosphodiester backbone missing a base. The site is called an AP site. In this case, it is missing a purine and is therefore an apurinic site. If it were missing a pyrimidine, it would be an apyrimidinic site. The next step in base excision repair involves AP endonucleases that specifically cleave the phosphodiester backbone and AP sites. The 3' end produced by the AP endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the AP site. DNA ligase then seals the remaining nick and the repair process is complete.
That was like a really helpful video. You should watch that before the survey session. But obviously it told you a little more than you needed to know about
Yeah.
those.
Doesn't have to be that
Specifically what
specific.
happens in what's happening in the case where it's not uracil and when we're changing things and those enzymes. So you just need to know that the AP endonucleases come in and they can recognize those sites. That's what those enzymes are doing. So any questions on that? That's a great video. I feel like we should send that video out. That's an awesome video. Okay, on to the next one. And we are going to compare this now with nucleotide excision repair, which repairs large lesions in the DNA. So remember the pyrimidine dimers, that's like a big deal. That's a big deal problem. And so we identified the lesion by a protein. You don't need to know the protein in this case. That has endonuclease activity. Endonuclease, we know it's going to cut, it's going to nick the DNA. Helicase removes the damaged strand, it gets degraded, and then the DNA polymerase comes in to fill the gap, and ligase comes in and seals it back up. So this seems very similar, but in this case, we're taking out a big chunk using DNA helicase. And then DNA polymerase and ligase are coming in and filling in the whole big chunk versus just like taking out one base, chopping a few of them, and filling it back up. Does that make sense? Okay, and now we have, what if you had a loss function in the NER mechanism? What do you think might happen? That would be bad. Yes, why would it be bad?
Can't fix the timing of dimers. So they would just stay covalently bonded to each other. And then, yeah,
And what does that do?
then they can't base pair.
Well, we mess up the DNA, like the form of the DNA altogether. So it's kind of one of those major things that if we're messing up that DNA, we mutated it really, really bad to the point that it's completely non-functional. Does that make sense? Okay.
Or also a quick video on NER.
Oh, we got another one? Yes.
Yeah.
I'm really glad that there were no weird emails.
Wow.
Just record it.
Oh yeah, just work hard.
Nucleotide excision
Oh
repair
no.
is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA,
You all need to
UVRP,
know this is the
UVRC,
protein.
and UVRD.
Memorize all
UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage, such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
I need to know.
Nucleotide excision
No,
repair.
not again. 2013. How do you think they hire people? How do you think they pick those voices? Nucleotide excision repair.
E. coli.
That's great visualization. You don't need to know all the enzymes, but that's I didn't know that the enzyme that comes in and cuts looks like that. But any questions on NER
No.
versus BER? Yeah. Double-stranded breaks. Spoiler alert, you break both strands. So this can happen because of UV again, and then ionization radiation. So UV causes a lot of issues, which is why we don't like UV and we wear sunscreen. Also can be unrepaired lesions, like we were talking about, issues with when our DNA proofreading or repair does not work. And there's 2 ways to repair this, and that's non-homologous end joining or homology-directed repair. So we're going to look at those. So non-homologous end joining, we identify the broken blunt ends of the DNA, we process them with endonuclease, and then we join them back together. So we take our blunt ends and we endonuclease so that now we're equal, and then we join them back together. There's no other complementary DNA that we repair this, that we do this when there's no other complementary DNA to come and fix this for us. Because obviously we don't need any DNA, we just cut it, stick it back together. So this is great if we don't have a homologous strand of DNA, then any double-stranded DNA break can be fixed, because double-stranded DNA breaks are definitely a problem. But the con is, because we use an endonuclease, we're losing nucleotides, right? So we chop these nucleotides. So we lose that, and we can lose genetic information doing that. So
Sorry, is it endo or exo? Because the slide says exo.
exo, I'm so sorry. Yeah, that's my dyslexia. The exonuclease. Yeah. There's a lot of words that are very similar. If you didn't see the UVRA, UVRB, UVRC, people suck in bio at naming. So does that make sense? Where we possibly could be losing some information, genetic information here, but we don't need a homologous strand of DNA. So this is more versatile. Versus,
I have a video on this, so we're gonna watch that first, and
Oh
then
my
we'll talk
gosh.
about the other one. I'm a visual learner. I like to see what I'm talking about.
I just like to hear myself talk.
Okay,
That's a joke.
sure.
I'm gonna remember every second of this video, and I'm gonna cry myself to sleep because of you. Anyone else see all the Taylor Swift video recommendations on the side?
DNA's role serves high importance, and yet it's vulnerable. Endogenous
This is worse.
I don't like this voice.
and exogenous factors such as ionizing
It's a British
radiation
child.
and
Can you pause it for just a
X-rays
second?
cause double-strand breaks. Oh, it's a double.
What horror film child did they come in to have record this video? I'm sorry, you can watch it, but like I'm upset.
It's a little girl question.
I'm so scared. Go ahead. Do we not want to watch it? This one.
It's all
No, just
right.
play.
Double break is formed. Within seconds, the Q heterodimer recognizes the break and joins
Heterodimer.
the DNA ends. Q's high affinity for DNA and abundance in the cell allows for its fast binding. It recruits the DNA protein kinase catalytic subunit. This relocates Q inwards so it can access the broken ends. Together, they form a synaptic complex called DNA PK, which bridges the broken ends together. The nucleus Artemis is sometimes recruited to process these ends, removing any damaged or incompatible nucleotides. In general, the process is little to no end processing. However, a small subset of breaks requires it for accurate repair. Two structurally similar proteins are recruited, XLCC4 and XLF. Their heads contain highly conserved regions which interact, creating a scaffold that forms a bridge between the DNA strands. The XLCC4 and XLF dimers alternate, forming a long, grooved, superhelical bundle of filaments. Only one here for simplicity. This bundle aligns the ends for ligation. Ligase 4 is recruited in a complex with the tail of XRCC4. It interacts with the two broken ends, undergoing a conformational change to encircle the DNA. Ligase 4 modifies the ends, ultimately forming a phosphodiester bond, ligating the ends together. Non-homologous end joining is error-prone due to its flexibility to directly ligate ends regardless of sequence and without the need of a homologous template strand.
I just feel like
So scary.
that's the kind of like, I think I just had a fever dream. Like I think someone just slipped me some acid or something. That was wild. I'm really glad you made us watch that, Chi-Chi. Does everyone feel like they better understand the concept after watching that fever dream of a video?
Okay.
I didn't like how detailed it was. You know, like little bumps on the proteins. Like
I don't know who made that, but I'm uncomfortable and I hate it. Okay. Do we have any questions on this before we move on to our comparison? Okay.
Oh, you gotta watch another video.
They better not be for the same company. Okay, and then homology directed repair is different because we have a homologous strand available, and we use that as a template to perfectly repair. And we use homologous recombination. And we prefer to use this because, as our creepy little girl said, the other one is error prone. This is not. You're not losing information. So con is the only downside is that we have to have another double strand version of the broken DNA. But we repair without any changes to post sequence. So this is just showing the difference between them. This is obviously simpler, but we can have our homologous recombination. This is where our mutation is. We use helicase to open it up. This is our homologous region. We come in, we repair it. No change in the DNA. Good. I'm sure Chi-Chi's gonna have another great video for us. Let's see. Do you not?
I don't like this.
So we're not watching it then. Is everyone okay with not watching another disturbing video? We good? Okay. You can search for one. So do we have any questions on the difference between these two? No, I think that video explained it so well. That I don't think you'll ever have a question again. Okay. Yeah, Artemis. Like, who named that protein? Okay. Chi-Chi. On to the next one.
What's that?
Oh, thanks for
I thought there were more slides. I was gonna ask that then, but on the quiz, I was kind of confused because if you have more, like if the cell is dividing more, would it involve homologous or non-homologous pair?
You're always going to use this if you have homologous double-stranded DNA. If you have another version of this double-stranded DNA, you're always going to want to use this. What specifically are you asking about? I don't know. Like, I don't.
She says,
Can I read the question?
doesn't give any context.
So the DSB repair pathway is more commonly used in rapidly dividing cells or cells that divide often. So this DS is this DS. So I think that this is the answer because, so the answer to that question, everyone, was homology directed repair, because this is what is being used most of the time. And if we're rapidly dividing, there's a good chance that if we're dividing a lot, there's a good chance that we have available.
On the slides, one of the lectures, she said like we use non-homologous more often, not body or something like that.
You know, I could not speak to that. I don't remember that. So I'm not sure. But basically, this is our preference for not losing DNA. Okay, well, maybe we can look into that. I'll try to find that slide and send out an email about it. Just because I don't want to be telling you something different, but this is definitely homology directed repair is our preference because we're not losing any information and it's less error prone.
But the question doesn't ask anything.
I'm just assuming that if we're rapidly dividing, my thought process is we're going to have double-stranded versions of this broken DNA if we're dividing because
in her like the last slide, she said majority of double-stranded breaks are non-homologous.
That's because human cells are not, they're usually, so if you remember from the cell cycle, most of our cells, they spend most of their time in interphase. We're not usually dividing. So in ones that are dividing a lot, this is what we're using because we have that double-stranded homologous DNA.
Do we just assume that there's a template?
When it's dividing, we're copying the DNA. Yeah. So normally we have two sets of DNA, right? So we have this extra homologous strand. So this is what we're going to use when we're dividing because we know when we're dividing, we're copying. So we have that other homologous strand. In cells that aren't dividing, it's not as likely that we have that strand. So the other way is going to be favored. Does that make sense? A little more?
Yeah.
It's definitely hard, but it's just like when you think about it, we don't have just like extraneous DNA floating around.
Yeah.
For this one, we need the exact DNA that matches where this mutation is happening. And you're going to find that when you're dividing because you have that copied DNA.
So if it's dividing a lot, it means there has to be another template.
Yeah, well, you're going to have duplicated DNA. If we're dividing a lot, that's what's happening. Versus an interphase, which is where most of the cells in our body are at all times, where we'd be very large people. We would just keep growing forever. We're normally going to use non-homologous because we don't just have extra double-stranded homologous DNA floating around. So that also makes a lot of sense as to why in S phase and there's a lot of repair mechanisms because we're making mutations and mistakes when we're using non-homologous repair, right?
Yeah, that makes sense.
So that kind of, we like this, but it's not as efficient and it's pickier. Or I guess it's more, efficient is not a great word, but it's not as picky. Does that make sense? Better? Yeah. Oh, look, I did something. Any other questions?
I'm trying to find this video.
She's still looking for that video, guys. You're gonna find a homology directed repair video.
This is weird.
How's everyone feeling? Any question on this topic? Any other topics? Are we just excited for a break because we're all drained?
Does anyone want to talk about?
Yay, Laura, for giving us Monday off. Woo! Love to hear it. I'm going home Saturday. You can go to the last slide, or end slide while you're waiting. Later, I have to split. It's funny.
Is that what's funny?
No. I thought that the informatics that Laura had on the slide Monday that she thought was a meme hilarious. That was a real knee slapper. Gigi, were you there?
No.
When Laura had it, she was like, look at this meme, and it was just like an informatic. It was just a picture of like what just like information it was.
I was like, that's not.
Do you know what you grading against?
I think that we just finished adding them today. I was doing that earlier. Michelle, we're working. They'll be put in by the end of the week, I believe. It's Thursday, bro.
She told us we have on Sunday, bro.
Yeah, it's Thursday.
Sunday's in last Sunday.
Do you know how long it takes to grade and tally up the exam?
Well, Emily, I actually don't. I have the privilege of not knowing that.
Okay, well,
Yeah.
let's have some patience because as much as we struggle, everyone else struggles and you don't understand you don't know what everyone else is going through. So I personally, for grading and adding up, I feel as
You
though
guys all did terrible.
Damn.
everyone failed. No, I feel as though it seemed to me that there was stronger performance on this. I don't know the average or anything. This is just what I've seen. Obviously, people still there.
Yeah, a lot of your-
But it felt like people knew what they were talking about a little more.
Your answers improved from first exam.
I definitely gave a lot more full concept questions. Okay, let's end the Zoom recording.
Okay.
Okay, bye everyone on Zoom. I'm so excited that that's gonna be on YouTube forever.
Oh my God.
Get me out of here.